In part 1 of Chapter 9, we discussed how you would take a DNA of unknown sequence, amplify it using polymerase chain reaction, and then use the chain terminator method to identify the sequence of that unknown DNA. So now we are going to turn our attention to how you might use that information. Once you have a DNA of known base nucleotide sequence, what do you do with that information? So here, we're going to jump right in and turn our attention to that question. The methods that we will talk about fall under the umbrella of recombinant DNA technology. First, before we really talk about this at all, I want to just give you a definition. What is recombinant DNA technology? Well, all this is a set of methods that makes it possible to isolate, amplify, and then modify specific DNA sequences. Recombinant DNA technology is often called molecular cloning or genetic engineering, so you'll hear it referred to by different names. But all this is a set of techniques that allows you to first isolate your DNA of interest and then amplify it, think polymerase chain reaction, and modify specific DNA sequences. So you presumably at this stage would know the DNA sequence of the thing that you have isolated and amplified using the methods that we had talked about last time. So now we're going to talk about, in this last bit here, how you might use that in a way to modify the sequences. So the general workflow is going to look something like this. And you'll see that we're going to walk through this overall workflow. But generally speaking, you would first start by obtaining a fragment of DNA of the appropriate size. So this would be a fragment of DNA that has been, say, cut out of the genome using restriction enzymes. So if you were to isolate genomic DNA and identify restriction enzyme sites, you could theoretically take that genomic material and just cut your DNA sequence of interest out of it. Alternatively, you could generate from genomic DNA using polymerase chain reaction fragments of DNA that you are interested in just simply by amplifying certain gene regions or certain DNA regions from genomic DNA. Or in the last option, you could just undergo chemical synthesis. There are ways to chemically synthesize DNA. And when I say this, I don't mean, you can't chemically synthesize the genome, but smaller DNA fragments, sure, you certainly can undergo chemical synthesis. So the first step really here is to obtain your isolated DNA sequence that you are interested in. The next step here is going to be incorporation of that DNA into another molecule of DNA known as a vector. So a vector is in a sense like a carrier of your DNA that you're interested in. So you take your vector, insert your foreign DNA into that vector to generate what is referred to as a plasmid. You'll see why you need to do this later on, but this is your next step. Then your vector plus the DNA of interest is going to be introduced into cells, and this is going to allow for the propagation of your plasmid, your vector plus DNA. So you are going to see that this approach is going to allow you to select for cells that have your plasmid. Overall, you're going to use the cells to amplify your plasmid, and this works, and you'll see why as we go on. And the cells that contain the desired DNA are going to be identified based on certain fitness advantages that the vector confers upon the cells. So we're going to walk through this, but this is the overall view, the overall workflow involved in the isolation and amplification of your DNA of interest. We are going to start here at the level of the plasmid. This is something that you're going to hear me refer to a lot going forward, so it makes sense to just start with a discussion of what a plasmid is. Well, plasmids are just circular DNA molecules that range from 1 to 200 kilobases, and these are commonly found in bacteria or yeast. They are always considered as extra chromosomal genetic elements. So if you look at an organism like a bacterium, you'll see that your plasmid is just a circular DNA that occurs outside of your chromosome. Outside of your bacterial DNA, your chromosomal DNA. So plasmids are readily transferable between bacteria, and you can see what is referred to as horizontal gene transfer, where plasmids are just passed around between bacteria, and this is why we refer to it as an extra chromosomal genetic element. Now, plasmids, in a sense, are referred to or could be thought of as molecular parasites. They themselves basically just hitch a ride. They move into the bacteria, and if the plasmid confers some sort of fitness advantage to the organism, they benefit the host, and then the host will propagate the plasmid, basically just spread it around. So these fitness advantages might include antibiotic resistance or thermotolerance. There are different fitness advantages that one could imagine, but in order for a plasmid to be propagated and really function successfully as a parasite, it would need to confer some sort of fitness advantage that allows the population of bacteria or organisms that have the plasmid to really outcompete those that do not. So when we think about plasmid DNA, there are different kinds of plasmids, and generally in the conversation that we are having now, we would refer to them based on a parameter that is referred to as the copy number. So the copy number tells us the number of copies of plasmid per cell or mass of cells. And this is important because there are plasmids that are referred to as high copy number plasmids, and there are also low copy number plasmids. In some cases, you'll see plasmids typically present in, say, hundreds of copies per cell, but others can be as high as 2,000 to 3,000. So you have low copy number plasmids and then high copy number plasmids. In the conversation that we are having here, this is important because knowledge of whether or not a plasmid is low or high copy number may be important when it comes to the consideration of the gene that you are trying to build upon, that you're trying to use in your work. So in some cases, as you will see later, there may be some mild toxicity associated with your efforts. In those cases, a low copy number plasmid may be more appropriate to minimize the toxicity of your work. Whereas in other cases, toxicity is not an issue, and so you can use high copy number plasmids, where basically you just want to make more of the thing that you're trying to make. So I'm being intentionally vague in my language there, but just know that plasmids can be described as high copy number or low copy number, and this is just simply a reference to the number of copies of your plasmid per cell or mass of cells. So what makes for a good plasmid? Well, in the wild, this conversation is very different than the one we are about to have. So the conversation that we are going to have here is going to focus on what is referred to as a cloning plasmid. So this is going to be a plasmid that is compatible with doing molecular cloning, and this is lab-based work. So just know that everything I'm about to tell you is not exactly translatable to the plasmid that you find in horizontal gene transfer in more native conditions. This is a lab-based, biotechnology-based conversation. So just know that. So from this point forward, this thing that I'm about to describe to you at this stage is a vector. So the vector here, this is going to be the carrier of your gene of interest or your foreign DNA of interest. So I'm about to describe to you the characteristics of a typical cloning vector, not actually a plasmid. This wording is a little bit misleading. So plasmid here is going to refer to my vector, which is shown to you, plus my gene or foreign DNA of interest. So the discussion I'm about to have is related to the typical cloning vector characteristics. So all of your cloning vectors, you want them to be relatively small. That's the first point here. This is important because the smaller the vector is, the more flexibility you have with regards to the gene or foreign DNA of interest that you're going to insert into your vector. You have to understand that in order to get your plasmid into cells, bigger is not better. So in this case, small plasmids are going to transform into cells with much higher efficiency relative to big plasmids. So big plasmids are not uptaken by cells quite as readily as small plasmids. So you want something small. Small is better here. The upper limit for size for a vector is going to be in the ballpark of about 10 kilobases. So that is really the upper limit. Okay, so here I'm showing you the Pt2 vector, which is 2.69 kilobases. So we are far away from that upper limit here. A typical cloning vector would also replicate easily. So once you get it into cells, you want the cells to be pretty readily able to replicate your cloning vector or plasmid. And this is good because then you basically just have to get one copy of your plasmid into a cell, and then the cell will start replicating it, especially if there's a fitness advantage. And you can then sort of reach your low or high copy number steady state condition. Okay, so the third point here, you want your vector to carry certain genes that confer antibiotic resistance or some sort of fitness advantage. So here I'm going to point you to certain features of this vector. So you'll see a bunch of stuff over here. You'll see each of these tick marks indicate restriction enzyme sites. So for example, this tick mark right here, if I treated the vector with NDE1, this is a restriction enzyme. NDE1 would come along, it would bind here, and it would then cleave, my vector. And so my circular DNA would no longer be circular. It would be an open linear DNA because I have treated my circular DNA with a restriction enzyme that cleaved the phosphodiester backbone and basically opened my circle into a linear structure. So you'll see restriction sites all over the place here. But you'll also see in colored regions certain what we refer to as cassettes. So here I have an ampicillin resistance cassette right over here. So ampicillin is a very common antibiotic, and for organisms that have this Pt2 vector with an intact ampicillin resistance cassette, those organisms should be resistant to ampicillin. So within this cassette, you'll find usually multiple genes that encode for enzymes that metabolize the antibiotic in question. And so by basically binding and metabolizing, degrading the antibiotic in question, there then is a resistance for the organism to that particular antibiotic. So I'm going to bring together 2 points here. I told you that you have restriction sites all over the place here, but you'll also notice that there are restriction sites right in the middle of this ampicillin resistance cassette. This is important because if I were to, say, treat this vector with any of these restriction enzymes that cleave right in the middle of this ampicillin resistance cassette, disrupt the continuity of the ampicillin resistance cassette, and then let's say I move that into an organism, I have basically destroyed the ampicillin resistance aspect of this vector. So I need this to be intact. I need to avoid, if I can, the treatment of this vector with any of these restriction enzymes. So I want that to be intact. Okay, so the fourth point here, the vector should be compatible with the carrying of a gene encoding for a protein of interest. Usually you are going to use a cloning plasmid to produce a protein of interest downstream. So that is exactly what you're using this for in most cases. So you'll notice you've got some stuff over here. So here you actually have what is referred to as the lac operon, and we won't talk a whole lot about that here, but in the middle of this is something referred to as a polylinker. So you're going to use knowledge of your restriction sites and actually cut over here, and via sticky-ended cloning, basically you're going to cut with a sticky-ended or with a restriction enzyme that generates sticky ends, you're going to be able to insert a gene of interest into this region, and your plasmid is then created. Your vector plus your gene of interest equals plasmid. And so you should have a region in your vector that has restriction sites that are compatible with the insertion via sticky-ended cloning of a gene of interest to generate your plasmid. Your vector should also be of a known copy number. That is very important. You want to know if you have a high copy number plasmid or a low copy number plasmid. And the last point here, I have hit on repeatedly thus far. You should have conveniently located sites for restriction enzymes, restriction endonucleases. That allows you then to manipulate the structure of your overall vector. Let's talk about the general workflow here in practice, as opposed to the more abstract ideas I've already presented. So we are going to start with two things. You're going to have a cloning vector, and this is a vector that fits the characteristics that I have just described to you. And you're also going to start, in most cases, with chromosomal DNA. However, this could be chemically synthesized DNA. It could be chromosomal DNA. The two of those can be used pretty interchangeably here. But let's just assume that we are starting with chromosomal DNA. So in that instance, you are going to need to do polymerase chain reaction. So why? What? Huh? What? Okay. So you are going to need to isolate some DNA region, a gene perhaps, of interest, and take that and insert it into your vector to make the overall plasmid that you're interested in. So if you happen to get lucky, you could find that you have restriction enzyme sites on either end of your gene. You could treat your chromosomal DNA with those restriction enzymes and cut it out. Cut out your gene of interest and then ligate that into a vector. Sure, you could do that, but that is very unlikely. So what many people do is they take their chromosomal DNA and they design PCR primers and actually use PCR to amplify that gene of interest. So then you have a population of chromosomal DNA plus your gene of interest, and you would have to do some DNA purification in order to separate the two, but that's pretty straightforward. So, okay, at this stage, you have your chromosomal DNA, okay? You've amplified as we talked about, so now you have this. And if you have put some sort of forethought into this, you would have designed your DNA fragments that result from these methods to have sticky ends. So that's what the structures here are indicative of. You have ends that are potentially compatible with annealing as opposed to blunt ends that are not so compatible with annealing. Now, in order for these to be useful, you need to cut your vector open. So you're going to basically treat your vector with a restriction enzyme that is going to open up the circle into a linear DNA with ends that are compatible, that are ready for annealing. So then you can anneal your DNA of interest into that region, and using an enzyme called DNA ligase will basically glue it shut. So once you anneal your fragments in, you don't have a continuous phosphodiester backbone. So DNA ligase will come in and basically synthesize that backbone and recreate the circle that you started with, but now your circle has your gene or foreign DNA inserted in. So now at this stage, you have a recombinant plasmid, not a vector. You actually have a plasmid. Okay, so now you may not have a boatload of plasmid. You may have just a very small amount. So what are you going to do? How are you going to use this thing? Well, what you're going to do is introduce your plasmid into a host cell. This could be yeast, this could be E. coli, this could be, pick your favorite organism. So at this stage, you have a true plasmid. You have chromosomal DNA, and then you have extra chromosomal DNA in the form of the plasmid. So what you're going to do is you're going to propagate that organism, that cell, with plasmid. And so now you're going to have a much larger volume of plasmid, or rather plasmid containing cells, than you started with. Some cells will have a single copy, others will have multiple copies. But the point here is that you go from a small amount of plasmid to a much larger amount of plasmid, and then you can isolate that plasmid from your cells if need be, and actually wind up with a higher concentration of plasmid than you started with. Now, one question that you may have at this point is, oh, well, why can't I just use PCR to propagate, to amplify the overall plasmid, and then maybe ligate the thing shut? And why can't I do that? Well, PCR is very effective for the amplification of short DNA regions, but not necessarily entire plasmids. Remember, plasmids can be very large. So PCR is not the most effective way to amplify plasmids. This really is. Basically transform, get your plasmid into a cell, and then let the cells do most of the work for you. This is just a much more effective and efficient way to go about this. As always, there is a very good video on the internet somewhere to explain these concepts. So here, I'm going to just show you a video that really conveys a really solid summary of plasmid cloning. So let me start this and just let you watch it.
A sequence of DNA from a complex mixture of different DNA sequences. In order to clone a fragment of DNA, we need to insert it into a vector, usually a highly modified phage or plasmid that can replicate in a host cell. Generally, a plasmid vector contains three elements: a cloning site where the foreign DNA fragment can be inserted, a drug resistance gene which destroys antibiotics, in this case ampicillin, to allow selective growth of the host cell, and a replication origin to allow the plasmid to replicate in the host cell. To insert foreign DNA, we must first use a restriction enzyme to cleave the vector at the cloning site. Here, EcoR1 cleaves the palindromic sequence GAATTC to produce single-stranded ends called sticky ends. These can hybridize with any piece of DNA that has also been cut with EcoR1. Foreign DNA containing the sequence we wish to clone is digested with EcoR1 and then mixed with the cleaved vector. The sticky ends of the foreign and plasmid DNA molecules hybridize and then are sealed into phosphodiester linkages by the enzyme DNA ligase, creating a recombinant plasmid. Each of these recombinant plasmids contains the inserted DNA fragment, an ampicillin resistance gene, and a replication origin. A pool or library of circular recombinant plasmids is thus created. Each plasmid carries a unique fragment of foreign DNA, as indicated by the colored arrows. Next, the host cells, E. coli bacteria, are added to the recombinant plasmids. The cells have been treated with calcium chloride to make them permeable to DNA molecules. Through a process called transformation, a few cells take up a recombinant plasmid, while most other cells do not. The bacterial cells are poured onto a plate of nutrient agar containing the antibiotic ampicillin. With ampicillin in the agar, only cells resistant to the drug can grow. At 37 degrees, the cells will grow and multiply. Because they cannot move on the agar, each will produce a separate colony of cells. The non-transformed cells lack the plasmid with the ampicillin resistance gene and die. Cells that have a plasmid are resistant to ampicillin and survive. The replication origin allows the plasmid to replicate by using the host cell's enzymes. Plasmid replication is independent of host cell division, but plasmids are distributed to each daughter cell when the host cell divides. As the plasmids replicate and the host cells multiply, the number of copies of the recombinant plasmid is greatly amplified. The multiple daughter cells form a colony or clone. Because all the host cells in a colony are derived from a single cell, they all contain copies of the same recombinant plasmid with its fragment of foreign DNA. A variety of assay methods can now be used on the bacterial colonies to determine which contains the particular DNA sequence we wish to isolate. A sequence of DNA from a complex mixture of different.
So the conversation that we were just having was related to propagation of a foreign DNA in a bacterial cell. But there are other ways to do this beyond just simply transformation methods as described. There are actually bacteriophage approaches where you can take a bacteriophage that has a foreign DNA loaded up in it, and it'll infect your bacterium, and then your DNA is propagated in that way. There are similar ways to do this in, say, eukaryotic systems like insect cells. So insect cells are very common. Think Drosophila are very common for the alternative protein expression systems for, say, eukaryotic proteins. In those cases, you might use something called a baculovirus as opposed to a bacteriophage. But here the concept is very similar. So in this instance, what you're going to do is you're going to start with phage DNA. And we're going to focus on a system using bacteriophage lambda. This is just an alternative cloning vector approach. Here, we are limited in terms of the DNA insert, the gene or foreign DNA that you're interested in. We are limited to inserts up to about 16 kilobases. Now, why is that? Well, if we look at the phage DNA, so this is the starting phage DNA, you'll see that it's about just shy of 50 kilobases long. Okay, there's a middle region that is not actually not required for lytic infection. So what we can do is treat that with a restriction enzyme that is going to cut out the region that is not required. Okay, so that then leaves you with fragments. These fragments are about 36 kilobases. These are also the fragments that are required for infection. But before you make it to the point of DNA packaging and all of that, what you can do is via sticky-ended cloning, you can anneal and ligate a foreign DNA fragment that is about 15 kilobases into it to make what is referred to as a chimeric DNA. So basically what you've done is cut out the bit of DNA that is not required for lytic infection, and you have pasted in a foreign DNA that you are interested in. You have annealed and ligated, and now you have a phage DNA that carries your DNA of interest but is of the appropriate length. Now, why am I focusing on length here? DNA is packaged in phage particles only if the length of the insert is such that the total length is 75 to 105% of that original 48.5 kilobase wild type genome size. Excuse me. So I need to be within 75 to 105% of the overall original length that I had. So phage vectors with foreign DNA inserts that are too small fail to propagate because they cannot form infectious phage particles. So you need to be roughly the same size of the original phage DNA. So if you have met that requirement, you can then see in vitro packaging of your DNA into an infectious phage particle, which can then go on and infect the carrier, the host organisms that you are interested in. Now, just keep in mind that your flexibility is not quite what it is for the transformation approach. So you are limited to a certain range of DNA sizes. So this doesn't really give you a whole lot of wiggle room in terms of how big or small you can go with your gene inserts. But it is certainly an approach that is an alternative to the transformation approach. You will see this very commonly used for eukaryotic protein expression. I mentioned the instance of using baculoviruses to infect insect cells earlier. This is very commonly used to express proteins in a eukaryotic expression system where you have the possibility for post-translational modifications. So sometimes that is required, and this is an approach that is amenable to doing that. At this stage, we are going to assume that somehow you got your plasmid into a cell. But we're going to focus on bacterial cells for this conversation, so please just take that into account. But the idea here is that once your plasmid makes it into a bacterium, you then plate it onto an agar plate and grow colonies as described earlier. You need some way to select for bacteria that contain your plasmid versus those that don't. So here, it is going to be essential to select only your host organisms that have been transformed and contain a properly constructed vector. So we're going to talk about two general approaches to do this: the antibiotic pressure, selective pressure, and a chromogenic approach. So we're going to talk about these separately now. The first approach that we are going to talk about here is that of using antibiotic pressure for selection. So the example that we are about to talk about is a pretty clever way to go about this. But, as you saw earlier, it is often as easy as growing your bacteria in the presence of the appropriate antibiotic, and then just basically letting all of your bacteria that don't have your plasmid die off as a consequence of the antibiotic. So those that grow would only grow because they are resistant to the antibiotic as a consequence of carrying the appropriate plasmid. Now, this is not always a foolproof, 100% successful way to go, because sometimes you will see that your gene, your foreign DNA, is either not fully inserted or is inserted awkwardly. So simply seeing growth of your bacteria in the presence of the appropriate antibiotic does not guarantee that your bacteria also have your gene of interest. You can actually see cases where, let's say, your ligation efficiency did not go 100%, it didn't work properly as you might have expected, and you wind up with a mixture of empty vector and vector plus your gene of interest. If you just said, oh, do my bacteria grow or do they not? you wouldn't be able to separate between those two cases. So here is a way to do that. So let's say we start with a certain type of vector referred to as PBR322. So the PBR322 vector actually has two different antibiotic resistance cassettes. There's an ampicillin resistance cassette and a tetracycline resistance cassette. You're going to treat that vector with PST1 restriction enzyme, restriction endonuclease. It is going to cut at a site that sits right in the middle of the ampicillin resistance cassette. So now I have linear vector. My vector is no longer circular, it's been cut open. I'm going to paste in, anneal in my foreign DNA, which is going to, via sticky-ended complementarity, insert right in the middle of that ampicillin resistance cassette. And then I'm going to treat with DNA ligase to glue it shut. And now I should have foreign DNA inserted properly in, but disrupted ampicillin resistance at the same time. So my mixture of ligated species in some cases may just have empty vector. That's what you see here. Maybe my DNA didn't ligate in or anneal in as it should have. In other cases, you might see weird cases where your DNA has been truncated. And you'll see here the only real case where the entire DNA went in is this example right here. So the point is that you have a mixture of different DNAs. Okay, so how do I know that everything worked? What do I do? Well, you're going to take this mixture and transform E. coli cells. So you're going to see some cells have plasmid, others don't. But for example, how do you at this stage differentiate the correctly annealed DNA, the correct arrangement as you want it, versus those that don't? So let's talk about that. So what you're going to do is you're going to take your mixture of transformed cells and plate it. You're going to plate onto basically an LB auger plate that at first contains tetracycline. So remember, we cut in the middle of the ampicillin resistance cassette, not the tetracycline cassette. So the tetracycline cassette should be intact. So if I have empty vector or vector that has my gene inserted in that ampicillin resistance cassette, both of those should grow. Those situations should be functionally equivalent at this stage. So as long as you have vector, whether or not it has foreign DNA present or not, as long as you have vector, you should see growth here. Now, what you're going to do next is how we're going to physically separate and identify vector versus vector plus gene, so vector versus plasmid. So you're going to take a colony, so take colonies that you might be interested in, and in a grid-like fashion, you're going to transfer them to a tetracycline control plate, and then a plate that contains ampicillin and tetracycline. So you will see that for cases where you have correctly inserted foreign DNA, correctly inserted gene, you will not see growth at the corresponding position on the on the plate with ampicillin and tetracycline. So for example, right here, you'll see there's a colony on the tetracycline control plate, but there is not on the ampicillin plus tetracycline plate. So that tells me I have a colony that contains my plasmid with gene inserted into the middle of the ampicillin resistance cassette. You'll see other cases where, like here for example, you see a colony on both plates. This would be an example of a bacterial colony that only has vector. So the vector should confer, the empty vector should confer both ampicillin and tetracycline resistance. So if you have a colony on both plates, you have just empty vector. If you have a colony on the tetracycline plate but not the ampicillin plus tetracycline plate, there you have the plasmid that you have tried to make intentionally. So you can use different experimental strategies to really build upon the fitness advantage that antibiotics confer in order to select and identify colonies that contain the plasmid that you are interested in. Another route is commonly used for large plasmids. So this is a route that really relies upon physical selection. So let's just walk through this. You'll see that we're going to refer to this generally as the chromogenic approach, but let's just start up here. So we're going to start with this back vector. So the back vector, it has an origin of replication. It has a chloramphenicol, that abbreviated CAM, resistance cassette. So chloramphenicol is your antibiotic. So you could use an antibiotic selection approach here too, but that's not what we're going to do. So you're also going to see that this vector contains a gene encoding for LAC-Z. So rather, let me rephrase that. It contains the LAC-Z, which actually encodes for the enzyme beta-galactosidase. So beta-galactosidase, it normally is an enzyme that cleaves lactose into constituent monosaccharides. We've talked about beta-galactosidase previously in this class. So this is going to be important. Keep that in mind. What we're going to do is treat with a restriction enzyme that is going to cut right in the middle of that LAC-Z gene. So now we have cut right in the middle of that LAC-Z gene, and we are going to anneal a large DNA fragment with the appropriate sticky ends to generate our recombinant plasmid. Now, this approach is pretty commonly used to insert large DNA fragments, not small ones. So you might have to resort to this if you have a large gene that you're interested in. Now, you're going to take this large plasmid and use a method called electroporation to get it into a bacterial cell. Electroporation is commonly needed to facilitate transformation. It's a more high efficiency way because remember, the bigger the plasmid, the lower the efficiency of transformation. So you have to use a more aggressive approach to get this into the bacterium in the first place. So now you should be able to take your mixture of bacteria that hopefully contain your plasmid and transform onto an agar plate. Okay, so you could do this with the chloramphenicol approach, but that doesn't tell you whether or not you have your DNA inserted in. So what we're going to do is take advantage of the activity of beta-galactosidase. So if you inserted your DNA into your vector successfully as shown, you should have disrupted beta-galactosidase activity. Okay, so this is important because we're going to use that. So what we're going to do is actually grow our colonies on an agar plate that contains something called X-gal. Okay, let me just move to the next slide and I will show you a little bit more. So this is X-gal. X-gal in its intact form is colorless, but when beta-galactosidase is active, beta-galactosidase will cleave X-gal into beta-D-galactose and this thing here, which is blue. So if you have active beta-galactosidase, you should see that your colonies are blue. If you do not have active beta-galactosidase, your colonies should be just clear, not clear, but not blue. Should be more of your normal colony color. So what you can do at that stage is take your plate and pick colonies on the basis of blue or not blue. And so you would choose the non-blue colonies for, if you wanted to select for the properly inserted DNA. You remember we inserted our DNA right in the middle of that LAC-Z gene. So if you had proper insertion, your colonies should not be blue because you should not have active beta-galactosidase. Now we're going to turn to a discussion of what to do with your plasmid once you actually have it in your bacteria and that you have selected for colonies that have your plasmid. So what do you do? Well, a very common thing to do is bacterial expression. So Bacteria, especially E. coli, remain the most common host for doing this. Now, before we can really talk about the process, let's talk about the vector itself. So most of your vectors, I told you earlier, have this so-called polylinker region where you have restriction sites that allow you to paste in a foreign DNA or a gene that you're interested in. But what I didn't tell you is how you would control the production of the protein that corresponds. So we're going to use something called induction. And just to go ahead and give you the punchline, over to the right-hand side, I'm showing you a gel. We have two lanes. There's the so-called uninduced lane and the induced lane. In the uninduced lane, you see all of your normal bacterial proteins, but in the induced lane, you see those same proteins, but an additional, what I'm going to refer to as a big fat band, and this big fat band is our induced protein. So I made this using a plasmid containing the gene sequence for the protein REC-A, and under conditions that I have control of, the bacteria started making this REC-A, and now with the induced protein sample, I can take that and subject it to protein purification and isolate my REC-A protein. So from the plasmid, I was able to make the corresponding protein. Now, how does this happen? Well, if we look at the vector, we have our polylinker region. So here, this is where your REC-A gene would be inserted. But upstream, you see there's a transcription termination sequence. So here I have a stop codon built in, and cool. Upstream, I have a ribosome binding site. But upstream of that, I have bacterial promoter and operator sequences. So I have my promoter operators basically tell me, can I go or not? Ribosome is going to bind right here. And from that, in my corresponding messenger RNA, my ribosome will bind and make the synthesize the protein that I'm interested in, and then stop where the transcription termination sequence is. So basically what I'm telling you is I have built-in features that allow for me to turn on via control of operator, basically the accessibility of my operator, my promoter, can turn on expression of my protein. Now, this is controlled in most cases by the presence of lactose. So lactose or a lactose analog is my inducer. So when I add in lactose, my cells know to basically turn on production of everything in here and start making my protein. Now, let me give you the actual description of what this looks like. Why was I making a fuss about lactose? Well, there's a very good reason. The promoter operator region that we are talking about is actually part of the lac operon. This is intentional. We have taken an operon and incorporated it into our vector that allows for us to control whether or not we make our protein or not. So normally what's going to happen is going to look something like this. So without any lactose present, it's going to look like this. There's this LAC-I gene that is upstream of my promoter and operator. This is going to be used to make an mRNA, which is going to be used to make a repressor protein. This is the LAC repressor. So normally, this repressor is going to bind to that promoter operator region. And binding of the repressor basically blocks anything downstream from happening. So when the LAC repressor is bound to the promoter operator region, you cannot make your protein. So this is the uninduced state that you see over here. Now, okay, let's say I want to make my protein. I want to control when my protein is expressed versus when it is not. What I'm going to do is I'm going to introduce lactose, or more commonly, a non-hydrolyzable lactose analog, IPTG. Okay, so I add IPTG or lactose, and that binds to my LAC repressor. It actually causes a conformational change in the LAC repressor that alters the affinity of that protein for that promoter operator region, which then induces it to dissociate from that region. So at this stage, that then allows for, basically my promoter operator is opened up and RNA polymerase can bind to my promoter operator region. RNA polymerase is going to start moving on down and synthesizing the downstream genes. So when I say downstream, this is transcription we were talking about. RNA polymerase is going to bind, and if I go back a slide, let me just move back a slide. RNA polymerase is going to bind in here, and then make all of the stuff in between. And because of the transcription terminator sequence, is going to stop here. So I have produced an mRNA that can be used for production of my protein of interest. And within that mRNA is a ribosome binding site. So this all centers on accessibility of RNA polymerase to my promoter operator region, which is controlled by whether or not that lac repressor is bound, which is also controlled by the presence of lactose or IPTG. So when I add IPTG, the lac repressor dissociates, my RNA polymerase can bind, I can start making mRNA transcripts, which can then be used to synthesize protein, hence my induced protein. So in the uninduced state, the lac repressor is bound. In the induced state, it is not bound. And under those conditions, I can make my protein via the path that I have just laid out for you. So everything we've talked about implied that I was inserting a wild type, normal, non-modified gene of interest into a vector to generate my plasmid. Sometimes I might want to alter certain amino acids or certain regions of my resulting induced protein downstream. So that could mean that I delete a domain, or I add an internal extension, or I make substitute one amino acid for another. So all of these mutations can be done pretty readily via a method referred to as site-directed mutagenesis. So site-directed mutagenesis works kind of like this. I have a recombinant vector, or let's just say my plasmid here. My gene of interest is right here. I have already inserted it in. I've made my plasmid like I wanted to. Cool. Everything is cool. So here, this would be my wild type, non-modified gene of interest. But let's say I want to insert a single site mutation. What I can do is treat my plasmid with an appropriate restriction enzyme, introduce nicks here that allow me to basically pull out a certain internal region of my gene, and then I can take a mutated synthetic DNA segment and introduce that in, and basically I've cut out the normal part and I've replaced it with a DNA fragment that has a single site mutation, and then I can ligate this shut and use the methods that I've talked about to select for the appropriate plasmid and then induce protein expression. So I could use this to induce protein expression and then purify my mutant protein and test the functional consequences via enzyme assays of that original mutation that we're talking about here. So that first method of site-directed mutagenesis is more or less the old school way to go about doing that. There's a more straightforward and really faster approach that is more commonly utilized now that builds upon polymerase chain reaction. So here we have our plasmid, and it is a double-strand plasmid. So what you need to do first is denature the plasmid to separate the two strands, and then you're going to anneal primers to the region of DNA that you are interested in. So these primers are going to prime to your gene of interest that you're interested in, but the primers should contain actually the mutation that you are interested in. So imagine your gene stretches a more broadly spanning region, and you anneal your primer only to the section that is going to include the mutation that you are interested in. So your primer is where your mutation is introduced. So you prime, and then you use PCR, polymerase chain reaction, to extend. So your polymerase is going to synthesize DNA around the ring. Cool. And then what you're going to do at this stage, you have a mixture of DNA with mutation and without. So at this stage, remember, you still have your parent DNA, but you also have a new stretch of newly synthesized DNA. Now, the difference between these two DNAs is it goes beyond simply having the mutation or not. If I were to take this as is and express protein from it, I would have a mixture of wild type and mutant protein. So I need to get rid of the wild type plasmid. Now, if we look at this and think about it, if we think back to restriction enzymes, our conversation related to restriction enzymes, I told you that E. coli methylates its DNA to protect against invasion from bacteriophages. So this is important because here, this parent DNA, it came from E. coli. It is methylated. The newly synthesized DNA has not yet been in E. coli and is not methylated. So what we're going to do is actually treat this mixture with a methylation-specific nuclease. It is going to bind only to plasmid that is methylated. It is then going to digest only methylated plasmid, which leaves only non-methylated plasmid. So at this stage, I have digested my parent DNA, and I am left only with plasmid that contains that mutation. So I can then ligate this shut, I can transform it into cells, and I can express via the lactose induction strategy that we just talked about, express the mutant protein of interest, and then I can purify it and test the effect of that mutation on the function of the enzyme. So now we're going to come back around to a topic that we talked about when we hit protein purification in Chapter 5. So here, I told you that proteins can be modified on the genetic level to have affinity tags that can make purification a little bit easier. So now I'm pointing you to this at the current time so that you can understand how this actually occurs, how you would do this. You have common tags that we've talked about before that allow for greater ease of purification, but now you should see that what you can do is using recombinant DNA methods on the level of the plasmid, you can actually insert these tags and genetically connect them to your gene of interest so that the protein that is expressed downstream actually contains your tag of interest. Now, there are a couple of things to note here. The first of which is that many vectors, many recombinant vectors that you can purchase commercially actually include a tag upstream of the insertion site for your gene of interest. So based on the restriction enzyme that you decide to use, you can actually insert your DNA so that the vector provides an N-terminal or C-terminal affinity tag, most often in the form of the His tag or GST or maltose binding protein. But the point is that you can actually buy your vector buy a vector that has been pre-designed to incorporate an affinity tag just simply based on where you insert your gene. Now, another thing to take note of is that many people, including myself, actually prefer to not permanently leave the affinity tag on. So, there are many instances where it is known that the presence of an affinity tag can influence the conformation of the resulting protein downstream. So, what many people like to do is actually genetically include a linker sequence between your affinity tag and your gene of interest that then allows for preferential cleavage of your affinity tag off of your protein at a later time. Once you have produced the protein and taken advantage of the affinity purification strategy, you can then just remove the affinity tag and purify it away. So as an example, let's look at a GST labeled protein just to illustrate this point. Okay, so here we have our gene for our target protein, and we also have a separate gene for GST. And using the recombinant methods, the restriction enzyme-based cloning that we have talked about, you can actually cut into your plasmid that has your target gene and paste in your GST gene, so that when you express the protein, once transcription occurs and downstream when protein expression occurs, you are going to have a population of expressed proteins that have both your protein and a GST protein fused together. Now, to build upon the idea of cleaving that off later that I just presented to you, some people, including myself again, will include a linker between these two proteins. So here on the genetic level, you would insert a linker sequence between your target gene and your GST gene, and that linker sequence would contain the binding site for certain proteases that are specific and can basically downstream after purification allow for you to cleave off your GST here. Now, this has to occur, the cleavage has to occur after the affinity purification step though. And just as a reminder, in this instance, your GST domain is going to bind specifically to your column material, and then you can use free glutathione or free ligand to elute off your protein and separate it from all of your impurities. So at this stage, you might consider cleaving off your tag and separating, using maybe size exclusion chromatography or something like that. So the point of all of this is for you to understand that genetic modifications have opened many doors that were not available prior to the advent of PCR. So we are able to do things now using genetic modifications that many years ago were just out of the realm of possibility, just unthinkable. So we can easily incorporate mutations. We can easily delete things or add in new domains. We can do enzyme engineering. And all of this is made possible by recombinant DNA methods.